Hello everyone, my name is Dr. Somnath Ghatak and I am the application specialist for AIIMS India. Today we will be seeing a confocal system for sure, that is the LSM 980 from AIIMS India. But in addition to LSM 980, we also have a dedicated hardware based super resolution, that is the Airyscan2. So let's see what the system has to offer. So first of all, this is Axio Observer 7 and this is a fully motorized inverted fluorescence microscope. Onto this a very high-end and sophisticated system, we also have LSM 980 scan head, and this LSM 980 scan head is further has an attachment in the form of a Airyscan2. So what is this Airyscan2 and how this Airyscan2 is going to offer you a super resolution image is what we are going to discuss in details. Okay, now that you have seen the system with all the hardware attachments, what actually is present in this configuration, then comes to what is the resolution you are going to get with a super resolution module, that is a dedicated Airyscan module. In a confocal system, you will be getting an XY resolution of something around 250 nanometers in XY, and in Z around 500 or even more than that nanometer resolution. But when you talk of Airyscan resolution, the Airyscan, obviously when we are claiming it as a dedicated super resolution module, so this attachment or this dedicated hardware will give you a resolution of 120 to 130 nanometer in XY, and in the Z it will give a 350 nanometer resolution. So which means that almost there is a jump of 100 nanometer difference from a standard confocal to a Airyscan resolution, and that is going to give you a more meaningful and a more resolved data. Okay, now what is the Airyscan, why it is giving this improved resolution, and what is the hardware behind it? So I'll just give a brief outline about how the Airyscan works. Now Airyscan, as you saw, it's a dedicated hardware for the detector. So basically you can take it as a detector, a separate detector, which works in combination with other set of detectors which are available in the system. Now this detector is unique in a way. This is not a regular PMT detector which you are having as a standard configuration with a confocal system. But on the other side, in Airyscan we basically have 32 elements which is in the shape of a hexagon, and each of this hexagon 32 put in a honeycomb structure, so that each of these 32 elements in a Airyscan individually generates one PSF. So instead of having one pinhole and one PSF corresponding to that pinhole, in Airyscan we can generate 32 different PSFs for the same plane of focus. Now what this attributes, this 32 attributes, it actually is able to differentiate different orders of light. It actually identifies which is the 0th order, 1st order, 2nd order, and eventually the 3rd order of the light or the frequencies which is actually detecting. Based on this, there is an algorithm called Shepard sum, and this algorithm attributes or actually accumulates all the information from 32 individual PSFs and attributes to the central element, which is the central PSF, and that is why we are able to achieve a super resolution data from a normal confocal image. So why it is different? Because it is not a regular PMT. Second, it works on 32 individual PSF, which is by the virtue of 32 separate elements. And third, you get a resolved, much better resolved data compared to a confocal system. Now this is partly a deconvolution algorithm, partly a Shepard sum algorithm, which works all together to give you a better resolved data. But there is always a dedicated hardware involved in it. Without the hardware, if you are just taking the raw image or the raw confocal data and you try to do a lot of algorithms for per se like a deconvolution algorithm and try to extrapolate the super resolution data, then that data is basically caused due to or extracted by pixel reassignment. There is no raw data collected is actually a confocal image. It is not better than that. But on the other hand, when I talk of Airyscan, by taking 32 different PSFs, we are not only actually enriching or collecting more raw data in order to come to a super resolution data, but also we are collecting individuals and we are also dividing them into different orders of light. So that also helps me in removing the non-significant or maybe the blurry light or the non-focused signal, which still is being collected to some extent in a normal confocal system. Thus, we are able to achieve a much sharper and clearer with a better resolved scientific data using the Airyscan2. So, to start with, a normal glass slide I have for the demo purpose. This glass slide I'll be using, I have used it for other like for normal system demonstration also, and with the same I will go step by step. First I will image it with a standard confocal image, and then we'll go to the Airyscan, where we will improve onto the resolution and compare the data of a normal confocal image with the Airyscan image. And then we have also one very unique feature here, that is called multiplexing. So in multiplexing, what we actually do is we increase or we gain the temporal resolution. So not only in X, Y and Z, but also in temporal resolution we enhance from a standard confocal system. Means within the same second we can acquire more number of frames which a confocal system can give you. So it is wonderful when you are doing a time-lapse experiment, when you are doing dynamic and I mean to say fast dynamic experiments, perfusion, diffusion experiments, where your system is or your sample is moving at a very faster rate, which a normal confocal is not able to capture. Then you can definitely use Airyscan along with the confocal attachment, and then you can have better temporal resolution. In addition, you also can go to a more in-depth information. So for low light signal achievement at a depth of somewhere around 70 to 100 micron, you can achieve it using a Airyscan, which is otherwise not possible with the standard confocal system. So let's see how you're going with the software and how the software is designed for Airyscan and for your use. Okay, so the guest user interface, which is basically the Zen platform again, and this is Zen 3.1 version. So this platform is the same platform which we used the last time, and in this platform, if you see this part, the basic workflow or the light direction or light path rather, for bright field or for transmitted light path, you have a halogen light source which goes through the condenser. I'll just make this portion a little bigger so that it is visible. So as you can see now, it is passing through the condenser, through the objectives, and then the filter. So in case of a transmitted light path, there will be no filter. You can change it to no position. There will be no filter here, and then you can view it through the eyepiece or the ocular. But when you're using a fluorescence, then I think the light source will be this one, which is HXP 120 volt is equivalent to 120 watt light source, metal halide. Then through the shutter, it goes to the filter. And in filter, you have options of green, red and blue. 3 filters are inbuilt here. So if I want to see any of this, I have to select the filter, and then from here the light will go through the objective to the sample. From the sample, an emission path will come back and then it will come to the eyepiece. So what I do is, if I open the shutter, you can see the light is coming, and from the filter this is becoming a blue light, which is the excitation light. And from the sample, emission will be there, and this is the green emission which is collected or which you are seeing it in the ocular. Yes. So till here, this is the overall. If you want to capture it through the camera, then you also have a camera mode. You have to just change the light path from the ocular to the camera mode, and then you can see the green light is now directed towards the camera for acquisition. So once this is done, you have focused the sample, which I have done. Then I'll go to the acquisition part. So you can see the acquisition, and this acquisition tab will be open. So in this acquisition, what we'll do is we will open the smart setup, and in the smart setup now, in addition to LSM, we also have a wide field. So if you want to image it with the camera, you can also design your tracks accordingly. But today we will be focusing more on LSM and super resolution. So under the LSM, if you see, these are the tracks. I'll just delete this. Under this LSM, you have now these 2 modes. So LSM mode or a Airyscan mode. So first we will see the LSM mode, which is the laser scanning microscope, that is the confocal mode, and then we will go on to the Airyscan mode. So once I have selected, this is a highlighted blue portion, and from this plus I will go in the die list. So this die list is open. I have in my sample DAPI, I have Alexa 488, I have Alexa 568. So these three dies I have selected, and then best signal, you see these three tracks will be created. I'll just say OK, and when I say OK, under the channel I have three tracks created, but you see it is written confocal mode. So now I am taking images in the confocal mode. So in confocal, first thing is, the laser. So corresponding to Alexa floor 568, you have 561. What is the laser power I am using it? So to start with, I will use a laser power of 2%, and then I will check the pinhole, which is 1 array unit or not, and then the master gain as 650 volt. If this all three is done, I am good to go. So which I will do it for the green channel also. I will start with 2 and 1 array unit and 650 volt. Same for DAPI. Also I'll start with 2% of the laser, 1 array unit here, and then the 650 is the master gain. And I'll change the color of this DAPI because I'll make it as blue. So now I'll check each of them. I'll go in the live mode here, and in the live mode, if I click, I will get the images. So this image looks good, but I have to check whether it is saturated or not. So I'll go in the range indicator, and then it looks pretty okay to me. I will increase the digital offset also, so that the background becomes much more, out of noise, or whatever noise is there in the background is removed. I'll slightly also check the focus. So for focus, I'll just see in which plane it is coming, and I'll try to focus on the best focal plane. So this gives me the best focal plane here in the red. Then you go to the green. So in green, I have this image which looks pretty good to me. You see the histogram here. Okay. Then in DAPI, so DAPI is too much, 2% laser is too high. I will reduce it to 1%. And 1% looks good. In the range indicator, they are good. But you can further reduce it to 0.6 because there are other areas which are also visible here. So, and also I can reduce this. You can see here, all of you, this is where it is also collecting some portion of the green in the DAPI. So I will reduce this so that I get, now you see my image is much more clearer, and I am not getting anything apart from DAPI. So once I have done this, I will close this, and I will select this three selected. I'll go to the snap. So when it is taking a snap, now the image is in the confocal pixelation, which is 1908 by 1908. The sampling is 1%, and then my frame is taking 28.16 seconds. Pixel dwell time is 1.1 microsecond. Here I am taking it little bit slow. I can take it faster also if I think my signal to noise ratio is not good, but in this case my image looks good because my background is very black, my signal and the contrast is good enough. So I am happy. I can make it faster also if I want to make it or if I want to increase the scan speed. Now once this is done, 3 color imaging is done, what I can do is I can view them individually. So I'll split here, and then you can see the red, green and the blue. So all the 3. So what is this? Red is basically the mito tracker. The red is mito tracker. So you can see the mitochondria here. This is the cytoskeleton of the cell. So it is Alexa floor 488, which is turning the green, and this is the nucleus. Okay. So you get this in a confocal mode. Okay. Okay. Now this was a confocal image. Now we'll move on to the super resolution part, which is the Airyscan part. Same. I'll go to the I'll just close this and make this little bigger so that it is visible. Now in this, a smart setup. In the smart setup, now instead of going in the LSM mode, I will go in the Airyscan mode. Now in the Airyscan mode, you see a triangle will come. So this triangle will give you the preferences, like you want to do it at the fastest speed, or you want to do it with best resolution possible, or you want to just capture in a Airyscan confocal mode. So that is, these three options are there. So I want resolution because that is why the super resolution or Airyscan is meant for, and then I have taken these three tracks, rest everything is same. I'll say okay. So if I say okay, now my three tracks are prepared, and these three tracks are in the SR mode, super resolution mode. So as I did in the confocal, I will also do it same. So in this, first is 561 laser, I will make it as 2%, and then you have other control as the master gain. So master gain is 850, keep it at 850 by default. Then the green, which is also 2%, and the DAPI, which is also, I'm making it as 2%, and let's see to start with, because this is the same percentage of the laser which we used it in the confocal mode. So here first we'll see one by one, and there is one more thing which you need to check here. So this is basically the Airyscan module. Now you see here, this Airyscan module will keep on adjusting. So what I'll do is, now if I see this module, so these are like hexagons which I said, so you have 32 as a hexagonal elements present here, and then they will align, and then based on that we'll get it. And always you see there is a Airyscan acquisition is optimally configured. You have to always check whether your configuration is optimally done or not, then only you'll get the best resolution. So once you do it, so what happens is, this portion, you see the Airyscan element is there, and out of this is the center hexagonal element, and until and unless, so this reassignment is being done in order to give this as the brightest spot. So once this is brightest spot, this entire from red it will turn to green, and then we're good to go. So we'll just quickly cross check with the focus also. So in the focus also, if you check, let's see that which is the best focal plane we are getting it for this image. And once you are in the best focal plane, we also have an option, like if you don't want to do this again, you can ask the system also to do it. I am doing it here, I am asking it to do it for me, so I can simply activate this part where I say activation adjust in live and continuous scans, and adjust in time series. So during time series also, when it is running for a longer period of time, you can also do this. Okay, so this will take not much time, but yes, it is taking some time. So we'll just quickly see how best we can align it, and then once it gets aligned, it has to turn green. So we'll just do it, or we'll just see till how the system automatically. Now see, it has become green now. So once it is green, I'll close it. Now this system is optically configured, you can see the green, and then I can check it for other also. So for, let's see, this is I'm getting for the green channel, and then for the blue. So blue is too high, 2% is too high, we'll make it as 0.5%, and in 0.5% we are still getting little more, so less than that, 0.4, and then we will stop it. So once these three are optimized, what we'll do, we are in super resolution. Now important thing is, you see the sampling here. So this sampling is 2x, so twice the sampling which we used to do in a confocal system, and that is why the pixel, you see the pixelation is 2244 by 2244. Here we see the pixel dwell time, and the frame rate is approximately 33 seconds, and this is the image size which you are getting it. So once you are done, we'll just take a snap, and then we'll let the system take images, first red, green, and then blue, similarly which we did in the confocal system. Okay, so red, blue, green, these three are done. With Airyscan image, what we need to do is we need to process this image, because now this raw image is basically from 32 elements you are getting a projected image, which we need to process and get one image. So here we will come to the processing. Under processing, this is Airyscan processing. I have to click here and then automatically these parameters will take. This is the image which I have taken input. I just have to say apply. So if I say apply, then it will do the processing and then the processed image. So this is, now you see, is a processed Airyscan image. So if I do this way now, and then you can find out what is the processed image looks like. Now in this case, so it also looks more or less, for me also it is like a confocal image, but then is it better than the confocal image? So this is our confocal image, this is our Airyscan image. Let's compare. So I'll remove the raw file which we took in the Airyscan, and the last two images are my, one is a confocal image, one is a Airyscan image. So I'll split it. This is my Airyscan image and I need a snap of 3897. Yes. So this is my confocal image, and once I compare, so you see there is a change in the field of view. Yes, of course. And then we'll say one by one. Let's see. We'll take this one, and then you see the difference here. Okay. So here, if you see this portion, there is some signal where you are able to see, but here it is more or less the background noise is much less, and you see much more clarity. So if both of them, you know, they are meant to look very similar, but you see the resolution here at point, they are much better and well resolved. Your background noise is very less, which you have here. So you see the background noise is high, but in this case your background noise is much less. Okay. So similarly, if we compare with the other green, okay, in green also you see the differences here, the fine differences which is visible. So moment I do it, this looks much more grainy. So you can see here this is much more grainy and pixelated, but here this is much finer, and we can also pick up fine informations which is otherwise lost here. So if it's the same area, if I pick it up, so this is, and this area is the same, and here also, so these fine structures are more or less, they are blurry and it is not very you know, understandable or resolved in a way. But here in the Airyscan module, it is much better resolved and we are getting a much finer details, which otherwise in a confocal is little bit compromised. Similarly, if we compare with the DAPI, so nucleus I am not doing it because nucleus is basically a counter stain. So this is how, if we compare this two, and in terms of like taking the images. Okay, so this being the confocal image, this being the Airyscan image, this is the gross differences or gross likely I've compared with a confocal image with a super resolution image. But then we'll do one Z-stack, and then we'll find out, convert into 3D, and then we'll see what is the comparison in a Z-stack. Okay, so now you see we have captured the Z-stack images. This entire experiment took approximately 8 minutes, and 8 minutes it had 39 slices in 3 tracks, and here with interval of 0.15 micron, that was the parameter which we used. So after processing we get this data, and then if we see in the Z position, so these are the gradual Z positions which we acquired, and so if we create a 3D out of it, in 3D visualization we will get this. If I remove the other settings, so now this is the image you are getting it in a 3D, where you can separate it out your only mitochondria. So this is your only mitochondria data, and you can also have only your cytoskeleton. So this is cytoskeleton data which you can see and you can visualize. You can also have the surface data. So this is basically the surface analysis. So if you are doing something, you can see the surface of each and every. On the top surface, it will not give you in depth. You can also have a transparency data where your background is black and you can see till what is in-depth imaging. For that it is a good one, but usually maximum intensity projection, where from different depth you can have these images, and also you can have a depth encoding. So if I do a depth encoding here, you can see this is a very fine sample of fine cell which has hardly a depth of 2 micron, and here the red to blue, red is towards the cover slip and blue is away from the cover slip, although it is not visibly very different here. Let's see if I add other tracks, then also you can see. So this is the depth encoding, but if you want to see the positioning of the 3 tracks, you can easily do it. Okay, so once we have done this, what we can see, so this is basically in the Z stack with the super resolution mode, but there is one more modality, which is the multiplex, where we are increasing the temporal resolution. So the same experiment, if we do it with a multiplex mode, let's see how much time does it take. In this normal super resolution mode, we were able to capture it approximately 8 minutes for 39 slices. Let's see, keeping all the parameters same, in a multiplex mode, how fast we can acquire this. So in the multiplex mode, what I did is I went to the multiplex mode, and the same number of frames as I said, I have kept it. And as you can see, now this is in multiplex mode, and you see the time. So what has happened, my frame rate now it is only 3 seconds, 3.6 seconds, and you see the sampling is still 2%, the pixelation is even 2936 by 2936, which is even more, but the rate with which it is acquiring, it is almost like 1 minute. So in 1 minute I am able to acquire, and it is almost about to complete in another 12 seconds, and the same number of slices, same number of images, I am getting it with a better resolution than confocal, but at a much faster rate. And see, as you can see, 3 colors imaging, 39 slices I acquired in almost in 1 minute. So if we see this image, let's go in the process. In Airyscan, we'll just process this image, and we'll see the quality of this image compared to the previous one which we acquired. So you see this processing is happening. It will take certain time, okay, and then it will show. So it will take few seconds basically to process this image, and then we will get a processed and Airyscan image in the multiplex mode. So the idea with a multiplex mode is you have your sample, but the sample is moving. The dynamics of the sample is very fast, where you want to have a better resolution, but at the same time, as your confocal is slow, you are not able to capture more number of frames within the same given time. So here you can capture more number of frames. Now this is my process data in the same given time, and at a and the resolution is also very better, very similar to a standard super resolution of Airyscan. But also, if you have to keep in mind that when you're increasing the speed, the resolution slightly compromises from 120 to 140 nanometer in XY. So if you see this image and project it, and you see this image also fairly looks like the previous image. So I am getting more or less a very similar image, but at a very faster rate. So if I compare this image along with the previous image, so this is my Airyscan image, yes. So if you see this Airyscan image, this one along with this one, so what I have is, so if we compare these two images, now you see in the comparison these images are very similar, but one is been acquired in almost like 8 minutes and one is acquired in 1 minute. So that is the difference in the temporal resolution. You can capture the more or less same resolution in a much faster time.